Okay, so I wanted to go over the research paper format for us. And so here is an example research paper that I've picked up, and I want to kind of highlight a couple of things here. So first of all, if you take a look at a research paper, you're going to basically see a couple of major sections. Normally, you'll see what is called an abstract. That's just kind of like this little piece right here. And what it is kind of like just like the headline of the paper itself. So it kind of gives you a really, really quick synopsis of what it is they're studying and what they got. So the results that they actually found. And so this is kind of helpful so you can screen through lots of papers and really quickly be able to figure out what they did, what they had, and what they got. So I'm not going to require you to write an abstract. The other main piece that you're going to see in a research paper is going to be the intro and background. This is kind of like your description of the content matter. And so here you can see a little bit of the intro itself. So you can see this one has a very small intro. It's mostly the results, but this is basically going to be your intro. Your intro should be a little bit bigger. And typically the intro should be cited. So it should have cited references in there. For instance, in this particular journal, the strategy for citation is to use numbers, and it depends on the publication as to how they do that. So you can see here you referred to a particular paper, items 1 through 3, and if you take a look at your table of cited references, you will be able to see here is your what. So they're referring to these references here. The reason why that's important is because if you happen to mention a piece of research that informed what you did, and I find that to be very interesting, then I can go to your references page and I can actually find this particular paper and I can read it for myself. And so it's an important, a very important piece of scientific literature. I will require you to cite your intro. And in the intro, basically, you're going to be talking a little bit about sort of the background. So the intro's job is to essentially give background biology, describe the biology. Of the topic with citations. Okay, so that's one thing that you guys need to practice, is to kind of set your reader up for and explaining to them exactly kind of what your field is and what your discipline is. The things we do and don't know, those are the sorts of things that you want to get across in your intro. The other main section that you're going to see in a paper is a methods section, so materials and methods. Oftentimes that comes after the intro and background, which oftentimes if you have a lot of informing research can be quite lengthy, and it's okay if it's lengthy. You want to make sure that you're communicating the nature of your topic to your reader. The methods oftentimes will come afterwards. You can see in this particular journal, their format prefers to see the results first. But if you scroll through this particular paper, you can see the methods way down here at the end. So here is your methods, and this basically is a cookbook description. Of everything they did. So I'm going to extend that down here. So all of this is just descriptions of their methods. That's all their methods. And so That's going to be an important component of a paper. So what did you do and how did you do it? That's basically what your methods are. So this is like a cookbook description. And I always tell my students that a good method section, I should be able to read it and I should be able to do the exact experiment that you did by just reading your methods. And so that has to be detailed. So when you're looking at the procedures you're doing in lab, then you want to make sure that you are providing the methods. Imagine explaining what you did and how you did it to somebody who's never taken this class before. You know, maybe Joe Average off of Spear or something like that. So that's going to be a piece of what you're going to do. And so that oftentimes is a big section. Now I'm going to skip down here to give myself some room. Number 4 is results. And this is basically you presenting your data. And you can present your data in graphs, in pictures. You can do all kinds of things. You can do statistics. You can do all kinds of presentation of data, lots of different ways to present your data. And so in your results, you want to be able to present your data in multiple formats. One of the important things about your results section, and this is the most important piece, is when you take a look at your results. So here you can see this particular lab's results. And so they did several different things here. There's a lot going on here in their results. Notice they're all graphical. So they do have a couple of line graphs. In this case, they were taking a look at a particular signal as a function of time in days. And so they were plotting data. And so you could see they're also plotting absorbance changes over time. In this case, they were using different types of parameters. And they did that with different types of mixtures. So this is absorbance of a different mixture of things that they were looking at. So this is their graphical data. So this column here, they've organized as their graphical data. In this column here is a lot of their pictorial data. So this is like microscopy and things like that. This is your observational data set up in pictures. Notice they try to quantify it as much as possible. They give us, in this case, a sense of scale. So we know exactly how big these things are that they're looking at. And so that kind of helps us to get a sense of perspective. They also, this is a type of observational data as well, C and then G. These are like what's referred to as gel images. And we'll do electrophoresis in lab toward the end of the lab. And so this is another type of data. And then they have the signal associated with lots of different types of situations. And you can see they've plotted many, many lines on the same graph. So you can compare the lines side by side. A lot of times people like that. The other thing about graph, and this is what you want to do. So what you want to do is you want to basically recapitulate the graphing. So you want to present your data in graphical form, with an observational form. But in all cases, notice there's a couple of things here that are true for all of this. In this, you have the graphs are well labeled. I can look at these graphs and tell what's going on. Okay, so your graphs have to be well labeled. If they're not, sorry about that. I just want to check to make sure that I'm still on. So your graphs have to be well labeled. The second thing you'll notice is they're well described. So I don't know what these people did, and I don't know what they're studying. But if you notice, in the legend, you have a very nice description of exactly what you're seeing in each image, and that's important. And they tell you even a lot of times a little bit of what they did. Like for instance, here they can see you're looking at phenylalanine and the aggregation and co-aggregation. So they're explaining to you what they're seeing. So they're trying to explain what it is that you're actually seeing in your data. And so you will also want to have well-described images, whether it's a graph or whether it is an observational piece of data. And so they have lots of different types, but notice they have basically those two different types of data. So you have graphical data that's well labeled, and that's well described. So I can read this and know exactly what's going on. And then they have observational data. In this case, they used microscopy. That's also well labeled, right? Notice what they used. They used little descriptors of telling me what I'm seeing in each single box. And it's also well described, right? So that's basically what this is. So these are electron micrographs of these particular images. So you can see they're showing us this data. They're showing us the discoveries that they basically found. And so that's their results. The important thing about the results, I want to put this up here. The results, they need to be descriptive and well labeled and clear, easy to read. So one of the litmus tests that I use for results is I should be able to see your graphs and your results, and I should be able to have a good idea of what you got and what you discovered without reading a single word of your paper. Your results should be able to stand on their own and be communicative on their own. And it's a process. You want to start working on that and kind of playing around with that. But that is a really, really important section. That's the heart and soul of a research paper. And the last part is the conclusions, or what we refer to in research as the discussion. This is basically where you make your conclusions and you form your hypothesis. So this is where you basically interpret your data and you explain what the data says and how you make your conclusions from that data. This is your justification, okay? So your discussion is your conclusions, but it's not just a conclusion. You have to justify and argue why your data leads you to that conclusion. Why should I agree with your conclusion? This is your chance to basically argue that your data leads to this conclusion. And that's what's happening in the discussion section. So here you can see, this is not part of the discussion. This is just more results that have been sort of littered throughout. So you can see they are showing different effects on different types of tissues in the body. And the discussion is explaining and interpreting all of that. So this is where you're making your claims. This is where you say, I think this is the way it works. And this is the reason why. And this is where you're interpreting your data and using your data to justify your interpretation. So then, what we have then in this particular case is this sort of multi-part format. For the pH experiment, what we're going to do is we're going to start off with our intro. I'm not going to ask you to write an abstract just yet. This one's just going to be the intro. I want you to do a good intro, a good background, a thorough background with citations. And the second assignment, you're going to be working on both your results, basically taking your results from fermentation and photosynthesis and packaging them into graphs or observations. And you're going to be setting up a results section that has well-labeled graphs and they're well described in the figure legends. And that will be the second piece. And then for also for that piece, you're going to do a discussion section. So basically, what are your conclusions from your graphical data and why, right? Make your justification. So the one thing I also want you to do on the first assignment, the PH assignment, along with the intro, is your methods. So give me your methods, explain your methods. So you do intro and methods, and then the second one for fermentation and photosynthesis, you're going to do results and discussion. And so those will be the two pieces. And then the third and final piece is going to be doing all these sections together for the crime scene series. Okay. So that's basically the format of a paper, the basic sections, and what I want you guys to do. If you have any questions, let me know, and I will help you guys through this. I'll also mention and describe it a little bit more in class as well.